the meiosis 1, where the homologs will separate, and then meiosis 2, where the chromatids separate. Okay, so cerevisia, in contrast to mammalian cells, the nuclear membrane does not break down. So everything is occurring within this nuclear membrane, which is, and then later you have formation of a spore wall around these structures here to form the four spores. Okay, in terms of meiotic division, so here you have the chromosomes that are unreplicated, replication and recombination. Then you have meiosis 1, where the homologs separate, meiosis 2, where the chromatids separate, and you get 4 different spores, each haploid. Okay, and the temporal class, so the class in temporal meaning time, the ones that are expressed early require IME1, middle, and the T80, mid, late, and late. It's not known what the transcription factor or factors are. Okay, so the present study used a microarray that includes 97% of the known or predicted genes of yeast to explore their expression as a function of time during meiosis and sporulation. Okay, and so how is this microarray constructed? And I want to point out that this study was basically the first time that a microarray was used. Well, I should say this is the second time, but one of the authors here, who was a graduate student at Stanford, was the one that designed the way of doing this in a simplified manner. Now they have become very sophisticated, the way they do these arrays. But so there were actually 2 graduate students that are responsible for this work. So what they did was you PCR each open reading frame. So there are 6,200 in yeast. And yet then you array on a microscope slide. Yeah, the one you use in the lab to look at anything. It's a simple microscope slide. And this is done with a machine that they design. You know, they have pins and they put the DNA. Then you isolate the mRNA from vegetative cells and you label them with a green fluorescent dye. And you isolate mRNA from cells undergoing sporulation and meiosis, and you label them with a red fluorescent dye. And I'll show you a diagram. So here. So first of all, you PCR amplify each of the open reading frames, and here's your slide, and then you spot each one. Okay, and you spot them, you know, it's more than once, you know, because you need to make sure that you have replicates. So each of them is going to be spotted, but on different. So let's say this is open reading frame 1, but you can find open reading frame 1 here, and you could find it here, just as replicates. Okay, so, and remember, you can put many of these in just a square centimeter in a microscope slide. Now you have vegetative cells, that means yeast growing in the rich medium, and you extract the RNA, and you label this DNA with a green fluorescent dye, side 3, during the process of reverse transcription. So what does that mean? You convert this RNA to DNA using reverse transcriptase. This is an enzyme that can copy RNA into DNA. And this comes from viruses, animal viruses. Okay, so you have the green, and now here you have the cells undergoing meiosis and sporulation. You isolate the RNA at different time points. So I'll show you how many time points this is. And then you label with a red dye, and you generate red DNA molecules, and then you pull this together and you mix it with this during the process of hybridization, and there are different washes, and then you scan the using a scanner called fluorescent scanner to see the colors that you obtain there, and you're going to get different shades of green, red, yellow, and as I will show you in a moment. Okay. So here is showing how to do this. For example, in mammalian cells, you have a normal cell, cancer cell. You take the mRNA, you convert it into cDNA, again using reverse transcriptase and labeling with a green dye, and then here from cancer cells. And then you mix this and you add it to this, again, it's going to be a, in this case, again, it's like a microscope slide. They're going to have, but in this case, you do not have the whole gene. You have simply oligonucleotides that represent each of the genes, let's say in the human genome, okay? So this one will represent, human gene 1, and then this here, gene 20,000. There are 20,000 genes in humans. So you mix this, and they are going to, and you use multiple different oligonucleotides representing each gene. So the idea is when you mix this, they will hybridize to these probes, okay? And then you scan to see what you get, and you will get these different colors here. It's, of course, in a simplified manner. So yellow means that you have equal expression of the gene. This represents one gene of interest in both types of cells, normal and cancer. So that means that there is nothing unusual about that gene. Green means that you only express it in these cells, but not in these cells. Okay? And red means that they are expressed here. It means they are upregulated in the cancer cell, and those are the genes that, of course, researchers will be interested in studying. So go back to yeast. So the changes in the concentration of each mRNA transcript were measured at 7 different time points after switching the cells to sporulation medium, meiosis and sporulation. I keep saying sporulation, but this should be meiosis and sporulation. And the data are presented in a tabular form. And the changes in the concentration of each mRNA transcript, again, are measured by a scanner. And then I'll show you how the rows represent individual genes, columns, time points, etc. Something like this. So this is just taken a little bit of a microarray. So here's 0 point. This is where they are growing simply vegetatively, and here is half an hour, 2 hours, 5, 7, 9, 11. These are the time points through which this was done. So remember, all of these represents time where you have to be isolating RNAs. I remember I was in the lab where the student was, and I remember the time she spent in the cold room, because these RNAs are isolated in the cold room at 4 degrees the entire day. Okay, so here is a representation where you can see that there are some shades of red. So here this gene, so each of these represents one gene. Okay, so, and these are the time points. And here you can see that this gene is expressed and is maintained, its expression constant, but then it starts going down. Here it starts a little bit and then increases more. And here this one increases here much later and then increases more. And this one appears to be repressed here, then it's about equal expression and then increases late. Okay, so this is basically how these are read. Okay, and The changes in this expression are not shown by numerical values, but mapping to a color scale. So here, black is a 1-to-1 ratio. This corresponds to something that is yellow, a spot that is yellow. And then you can see the more intense red means higher expression as opposed to this that is a little darker red. And the same with the green. So the brighter the green, the more repressed or less expression of that gene compared to one that is darker green here. So that is to show you the scale. Okay. So the pattern of expression was independent, given that this was the first time this was done, and normally too, people always try to use an independent confirmation. So the pattern of expression was independently confirmed by northern blot analysis. So what is northern analysis? This involves fractionation of total RNA or mRNAs, depending on what you're doing, on a matrix. Usually this is a denaturing agarose gel, sorry for the misspelling there. And this is, then you transfer, I'll show you a diagram, the fractionated RNAs to a membrane, and then you use a radioactively labeled probe to detect a specific mRNA. This, of course, is done one gene at a time, so this is very time consuming, whereas a microarray, you are examining, in the case of yeast, 60 to 100 genes in one experiment. Okay? That's the big improvement in the impact of this technique. So what is northern? So a northern blot have RNA is fractionated on an agarose gel that is denaturing. So denaturing because RNA forms secondary structures, and you don't want that. You want the DNA to be linear. So you get kind of a smear. It's not like you can detect bands. And then what you do is you kind of sandwich this gel and put a membrane on top of the gel and make this sandwich with filter papers. And then you transfer, so you push a buffer to go through here, and that will cause the transfer of the DNA onto a nitrocellulose membrane. The RNA will bind to the nitrocellulose membrane, and you take that membrane and then you put it in a solution that has radioactively labeled probe for a gene of interest, and then you expose this to detect what bands are observed. And here is the X-ray film and the autoradiogram. So these bands here, these are the markers, and here is a band showing of all these RNAs that you loaded where your gene of interest is. You know, maybe people were looking at liver tissue, and brain tissue, and whatever. Okay, so that's northern. Okay, so here is showing northern. So northern for these 4 classes of genes. So these are typical genes. So this is an early gene. So you have a 0 time, meaning at the beginning of the experiment. You have basically nothing, and then you start seeing 2, the appearance of a strong band that increases but maintains more or less the same. And then this other gene that is, so this would be a typical early gene. This is a middle gene where the appearance of the RNA is 5 hours into the process, and then it increases. You know, you can see an increase the amount of the RNA, and this can be quantitative. Then you have a typical mid-late gene where you see the appearance of the RNA at 7 hours, and then dramatic increase later on. And then a late gene, typical late gene, where you have appearance here a little bit and then much later, okay? So this is by Northern analysis, but remember, for each of these, this is one gene at a time. Each of these represents a labeling with this probe. These are done independently, which is very time consuming. This is a very time consuming process. Okay, so when people did the analysis of the 6,200 protein coding genes, more than 1,000 showed significant changes in mRNA levels.